Click for IB Biology. Here we're looking at part 2 of the DNA mechanism of replication as appropriate to students taking the higher level course. There's a blank PDF of this presentation down in the description that you can annotate with your own notes. There's a 10-question quick quiz, and then there are channel links at the end to further aspects of this part of the syllabus. So let's recap. We are operating at the replication fork in a replication bubble. The helicase enzyme has broken the complementary base pairing between the leading template and the lagging template, and the nucleotides are beginning to complementary base pair onto both the leading template and the lagging template as shown in the diagram. And as was previously introduced, the first nucleotides to actually add to the templates are in fact RNA nucleotides added by DNA Primase. So this creates the so-called primers. Now these primers are what allows the polymerase 3, DNA polymerase 3, that's what allows the polymerase 3 to begin its enzyme action, and it can't do that unless it begins with a section of RNA nucleotides. Let's look at the process in more detail to understand why it is that the polymerase 3 on the leading strand is operating in this direction, whereas on the lagging strand the DNA polymerase is operating away from the replication fork in this direction. So let's take a look at that problem. And we go back to the early stages of the action of the helicase, in this case only actually having broken apart two of the nucleotide pairs of the templates, which are shown here in blue and red. And already we have the complementary base pairing of the new nucleotides in position, ready to be polymerized in their sugar phosphate backbone. Now the explanation of this polymerization of the sugar phosphate backbone on the new strands is often abbreviated simply to 5' added to 3'. This abbreviation hides a number of key points that the student needs to comprehend. So let me give you those. First of all, we're referring to the new strands. So that's both the leading strand here and the lagging strand there. So the discussion is about the new strands. Now the 5' is referring to the 5' carbon on the second nucleotide. I'll call it nucleo for short. And the 3' is referring to the first nucleotide. And between the two, what is being formed is the phosphodiester bond. So let's take a look at that on the leading strand to begin with. And so here we have the first nucleotide. So we need to identify the 3' carbon, and there it is there. And then we need to identify the 5' carbon on the second nucleotide, which is in this position here. So there's the 5' there, and there's the 3' there. And so the DNA polymerase will then add the bond through the phosphate, which is shown in green here, back to the 3' carbon. So we've added 5' to 3' through the phosphate and satisfied all the conditions necessary in our description. Let's take a look at the lagging strand. So the lagging strand template is here, the lagging strand is here. Let's identify the 3' on the lagging strand. This is here. And the 5' on the second nucleotide is all the way up here. And so this is not the position required by the active site of DNA polymerase 3. And therefore it cannot add the nucleotides of the first and second together on the lagging strand. So how is this all solved? Let's move on. Looking at the leading strand again. So we've got the leading strand template here. Here's the construction of the leading strand with its RNA nucleotide to begin with. There's number 2. So we've already created the first phosphodiester bond here. And then 5' here would be added to the 3' there. That would be the second phosphodiester bond. And then, of course, this is repeated for the next, for 4 to 3. So that would be the 5' here and the 3' there. And so the polymerase can maintain its operation following immediately behind the helicase. So we can identify at the replication fork where we have the helicase and the polymerase 3 operating closely together. This is the leading strand, and we continuously synthesize the chain in one smooth movement. But let's look at the lagging strand. Because the enzyme cannot attach the first and second nucleotides together, instead we have to add the RNA primers higher up the lagging strand towards the replication fork. And the polymerase will actually initiate its action in this location here. And will add the 5' on the second nucleotide to the 3' on the first nucleotide. So that's 3' there. And so we will create the bond here. Now the polymerase then needs to move, in our diagram, down the backbone, and will then take 3' is here, 5' is here, and so we can create the bond here. So you can see that the polymerase 3 is operating down the chain towards the beginning, like this. If we stand back from this detail and look at a larger overview, what we see in our model is the continuous synthesis of the new strand, the leading strand, here on the left-hand side. But on the right-hand side, the strand is being synthesized in a number of short sections. And these sections, often referred to as fragments, are called Okazaki fragments. What then happens is that we need to remove the primers, which, remember, are RNA nucleotides on the leading strand, and this is completed by DNA polymerase. The polymerase removes the RNA nucleotides and replaces them with DNA nucleotides. But then a second enzyme, DNA Ligase, completes the sugar phosphate backbone in that one location just there on the leading strand. If we look at the lagging strand, then it's necessary for the primers to be removed in a number of positions by polymerase 1, and then for the ligase enzyme to be operating in multiple locations, joining the sugar phosphate backbone of the lagging strand, so it gives us one continuous polynucleotide chain. In this way, the two sides of the molecule have been replicated. Occasionally, errors occur in this replication process, and so we'll now look at the DNA proofreading mechanism, and then there are links to the playlist showing you other considerations on the course, such as the applications of DNA technologies.